My name is Max Berner, and today we'll be going over the effect of PTU on the development of zebrafish embryo. So what is the problem? There's little understood about the process melanogenesis and how to regulate it. This process is responsible for making melanin, a common pigment found in organisms. A notable characteristic of PTU, the drug we used, is that it inhibits melanogenesis. Ideally, learning more about PTU can act as a gateway to help us learn more about melanogenesis to solve the problems we face surrounding that. The goal of this experiment was to learn more about PTU and its side effects so that it can be used more effectively to serve that purpose. PTU inhibits melanogenesis by inhibiting tyrosinase, an enzyme essential to the process. Without functioning tyrosinase, an organism cannot produce melanin. Albinism is a genetic disorder that exemplifies this. We are aiming to map out the side effects of PTU. By doing so, we can learn how to safely and effectively use PTU to create a baseline organism that simulates albinism, which is extremely helpful for testing out experimental treatments. But why should we care? While albinism could be strictly seen as a change in appearance, individuals with albinism face more issues than what are apparent. Melanin, like all things in our body, serves a purpose. It provides protection from radiation coming from the sun through UV rays. Individuals with albinism don't have this layer of protection, making them much more vulnerable to skin cancers. Additionally, due to their appearance, individuals with albinism face extreme prejudice in some cultures. An experiment done in 2005 confirmed that tyrosinase experiences a loss in function and an inhibition by PTU through a covalent bond that forms between the two of them. Another experiment done in 2001 notes that PTU doesn't remove pigment that is already produced, so it needs to be introduced in the embryonic period. Additionally, another experiment determined that when melanogenesis is disrupted, hypo and hyperpigmentation occur. An example of hypopigmentation would be albinism. The results of these three experiments suggest that when we introduce PTU in the embryonic period, the developing organism will turn out albino. So we designed a lab to answer, if PTU is introduced in the embryonic period, how will its presence affect the development of zebrafish embryo? Our independent variable was the presence of PTU. We referenced a lethal concentration graph on the right to determine our concentration of PTU, but we were also limited by our available resources. Dependent variables we decided to measure were the pigmentation percentage, hatching percentage, and mortality rate of the populations. In order to make this data quantifiable, we had to turn an observation into a number. To keep this uniform, we defined pigmented fish as a fish containing significant dark coloring disregarding its eyes or yolk. And a hatch fish we defined as a fish with its tail completely outside of the eggshell. Our control group was raised in an untampered instant ocean solution, which is a solution specifically made to accommodate for organisms like zebrafish. Since we were measuring three different dependent variables, we had to test three different hypotheses. Due to the inhibition of tyrosinase and reduced production of melanin, we hypothesized that the pigmentation percentage would decrease in populations exposed to PTU. And due to underlying effects of PTU, or side effects due to a disrupted melanogenesis process, populations exposed to PTU were hypothesized to hatch less and die in greater numbers. Our materials included zebrafish received from the UWM School of Freshwater Sciences, just a couple hours post-fertilization. We also used a 12-well plate to isolate our populations, an instant ocean solution to raise our embryos in, a PTU solution to expose the experimental groups, a stereo microscope, thin and wide nose pipettes to transport the solution and the eggs, and an incubator to keep the eggs at a constant temperature of 28.5 degrees Celsius overnight. We designed an experiment as our means to solve our research problem. Referencing the 12-well plate on the right, we decided to split up the populations of zebrafish by column. The first column was our control group, and our second column was exposed to PTU through its concentration. Our experiment did not require an IRB because we did not use human patients during our research, as we strictly used zebrafish. Our first step was to create the two solutions we would be using throughout our experiment. We used our instant ocean solution to dilute our PTU solution into 40 milliliters of a 0.005% PTU instant ocean solution. We then labeled the first column of wells as a control and our second column of wells as PTU. We inserted roughly 8 zebrafish eggs into each well using the wide pipettes. Each day we would replace the older solution in each well with a fresh solution of its designated type, determined by the label of the well, to remove waste. Also each day we recorded the pigmented fish, alive fish, and hatched fish in each well. We also removed the dead fish. Each night we would place the zebrafish in an incubator. Quantifiable data was collected by categorizing each zebrafish with the applying qualities: alive, hatched, and pigmented. Analysis was conducted by comparing the differences in those qualities between the two groups, the PTU group and the control group, and determining the trend statements. We chose an unpaired t-test as our statistical analysis because we were comparing two independent groups. The figure in the top right was categorized as pigmented, alive, and unhatched, and it was found in a well of a control group. The figure in the bottom right was categorized as not pigmented, alive, and unhatched, and it was found in a well of a PTU exposed group. Each day we followed a simple procedure and repeated it until 96 hours post-fertilization. We recorded our data daily for 4 days after fertilization. Each day we would record our data, refresh the wells that the zebrafish were in, and then put the zebrafish in the incubator overnight until we got them out the next day to repeat the same process. We observed a very distinct break around 48 hours post-fertilization, when the control group was progressing normally and hatching and developing pigmentation, while the PTU exposed group weren't hatching and they weren't developing any pigmentation. And that trend continued until 96 hours post-fertilization. As you can see in Figure 11, as time progressed, both groups suffered a decrease in survival rate. The PTU exposed groups felt a more drastic decline in their survival rate, with none of them surviving at 96 hours post-fertilization. This supported our hypothesis. However, although this difference may seem drastic, it was considered not statistically significant in our unpaired t-test by a very small margin. We think that if more trials were conducted, this trend would continue to show and that this would be considered statistically significant. The difference between the pigmentation was much more drastic, with all of the fish developing pigmentation in the control group, while none of the fish in the PTU exposed group developed pigmentation. This supported our hypothesis, and this was considered extremely statistically significant. Similar to the pigmentation percentage, there was a drastic difference in the hatched percentage. By 96 hours, all of the fish in the control group were hatched, but none of the fish in the PTU exposed group ever hatched. This supported our hypothesis and was also considered extremely statistically significant in an unpaired t-test. As stated before, we used an unpaired t-test to determine statistical significance because there were two independent groups, the PTU exposed group and the control group. The difference between our data for hatched percentage and pigmentation percentage between the two groups was considered extremely statistically significant. However, our data for survival rate was considered not statistically significant by a slight margin. The trends in our data supported all three of our hypotheses. We determined that PTU does have a harmful effect on developing embryo by disrupting their developmental process and lowering their survival rate. Although we do not know the exact cause behind it, we do know that PTU is causing an effect that is hindering the development of this organism. We also confirmed that PTU does inhibit melanogenesis, and that may hold a connection to why this organism's development is disrupted. The two biggest limitations to our experiment were our lack of trials and potential error. Due to a lack of resources, we didn't have enough PTU to expose fish at different levels of concentration. This limits the value of our data, as we only compared two different groups. Overall, we need to conduct more research and more trials to map out the effects of PTU and the extent of these effects. This will help us more effectively use it as a tool for research in regulating melanogenesis. Thank you for watching.